We are talking about the cell cycle in this particular chapter. This is chapter 12 of your textbook. And there are several things that are key with regard to, and I'm sorry I moved on from the cover or from the title slide a little too fast. We'll actually bring that back. You'll see this image again when we talk about the cycle of the cell. And many times when we talk about the cycle of the cell, and I didn't mean to boo-boo there, is you'll talk about it in its phases, or you'll talk about what the cell is actually doing and attach that to a portion of this circle. And then this cycle circle. And at each time there are checkpoints. These checkpoints are ways that are governors that make sure that this cell is actually still functioning normally. And if you have a cell that's not functioning normally, maybe because it's too old or so it has lost its viability, then other things might happen. It might have to need to be destroyed and replaced. But then moving on is we have this theory that all cells come from other cells. So every cell is from another cell. And so the only way that's going to happen is if we have a way to reproduce. So there is an overarching premise that the ability of an organism to produce more of their own kind best distinguishes living from non-living matter, this ability to reproduce. And unfortunately, that is simplistic because that has kind of been turned on its head with regard to viruses and other things that have been discovered to be able to replicate themselves in some capacity. The ability for life to continue is based on reproduction of cells. So cells are retired, you might say. They need to be replaced. And that replacement is what we call cell division. The cell division is basically what we're talking about here. Even unicellular organisms divide, is the division of one cell reproducing the entire organism. So what you have here, one of the reasons why that's even added is you have an image of a unicellular organism, and it's somewhat artistically enhanced, I might say, because you can see each of the little pieces of that unicellular organism. This particular organism is in the later stage of telekinesis or telekinesis and getting ready to actually separate into two daughter cells. Multicellular organisms depend on cell division for many things other than just replicating themselves. They depend on it for development of gametes, which are fertilized, are the ability to make more fertilized cells. They depend on it for the fertilized cell. They depend on it for growth, for repair. The cell division is very much part of the cell's job, its cycle, in the life of forming a cell from its own division. And what you have here are depictions of various functions of cells, reproduction, and we're reproducing a similar cell, or growth. What you can think of more tissue, where you have cells and they are attached to one another, and so we're developing tissues into organs. And then also, if a generally the cells, particularly in your body, have a lifespan, and when that lifespan is met, that particular cell needs to be replaced for many reasons, because that cell has a function, and also because that cell is taking up space. So the actual ability for an organ or an organ system or an organism to do and maintain itself in homeostasis is dependent on each individual cell. Maybe one or two cells can be lost, but cells take up mass in the body, and they also work in conjunction with other cells to perform specific tasks, so they need to be replaced. There are two basic cycles that we will be looking at: mitosis and meiosis. And this idea of daughter cells. So most division results in daughter cells with identical genetic information, or DNA, and that's mitosis. So in the human, we have 46 chromosomes, 23 pair, depending on how you talk about them. And the soma cells, your body cells, when they replicate, that's what we consider mitosis, where you have these 23 pair or 46 chromosomes that you have a exact copy of the cell before. And then the exception is meiosis. And that's a special type of division that produces the gametes, the sperm and the egg. We consider sperm the male gamete, the egg the female gamete. And the thing that happens with regard to meiosis that is unique, and we'll talk about that quite a bit, is this is where you have the ability to have thousands and trillions of combinations of the DNA. So you get this genetic variation. And a couple of things that are being brought up here that we want to point out, because we'll point them out again later on, is this idea of N, this 2N, this diploid and haploid with regard to cells. So if you have something that's 2N, you're thinking of it as diploid. It has a double complement of the chromosomal pairs, and in mitosis, that's a chromosomal pair that was received from the sperm, and a chromosomal piece of the pair, I guess you might say, from the female. So you have 2N, you have a pair, and then in meiosis, which I will probably say meiosis over and over again. It's actually meiosis and mitosis, but I'm trying to keep it very, very straight for you. At the end of meiosis cell division, you have a single chromatid, or a single chromosome, I guess you might say, I'm sorry, from each of the 23 pair sets. So you have 23 single chromosomes waiting to meet up with another 23 single chromosomes to start that pair up again. With regard to some terms, all the DNA in a cell, in an individual cell of an individual species, constitutes the cell's genome. And there's much discussion about genome and using genome and genome data for various different studies. But so we have a human genome that is our sum total of all of our DNA, and then say a butterfly has their own, and a fly has their own, and a monkey has their own. A genome can be a single DNA molecule that, if that makes up the full complement of the genome, thinking of prokaryotic cells or something simple, or a number of DNA molecules, as in most eukaryotic cells. They are packaged. DNA is packaged in chromosomes. And the DNA is a double helix, as you can see at the top of this image. That double helix is base pairs of nucleotides. Those that DNA is then spiraled and wrapped around these proteins called histones. And then you have them coiled into chromatid fibers, and then after that, further condensation, and then that condensation is what we then can visually see under the microscope as a condensed chromatin, which we call a chromosome. And so in the eukaryotic cell, chromosomes spend most of their time consisting of chromatid, so spend most of their time consisting of this fibrous material that's wrapped around histones and just in the nucleus itself. When the cell is ready to divide for cell division, then that chromatin is going to condense. It just makes it easier to move around. I'm sure there are other reasons, but when we look through the whole process, it's easier to move it if it's condensed. Whereas on the other hand, it's easier for proteins to be created out of bits of the genome if it is not condensed. So both of these are done for ease of the cell itself and doing what it needs to do. And then every species has their own characteristic number of chromosomes and also their sequencing. Now, you've probably heard a lot about the fact that the sequencing doesn't shift dramatically, or the amount of DNA doesn't shift dramatically, say between a human and a gibbon or a human and an orangutan, but each has their own unique characteristic number and sequencing of DNA. And then soma cells, as we've talked about, there's that term soma. Soma means body, have two of each. So that is because you get one from the male gamete, one from the female gamete. They get together into the zygote, and then every cell after that zygote replicates with that set of chromosomes. When the cell is getting ready for division, DNA is replicated. It has to be replicated because what we're going to do is create two daughter cells. Those two daughter cells are going to be identical. The first thing that makes them identical, they're going to be identical from every aspect, but the thing that makes them identical first is the replication of the DNA. Duplicate chromosomes has two sister chromatids, which are copies of the original chromosome. So sister chromatids are exact copies, and which are going to be able to separate during cell division. And remember, you have 23 pair of these. So each of the sister chromatids that is its exact copy are actually attached to one another at a narrow waist we call the centromere. So the narrow waist of the duplicated chromosome where these two chromatids are most closely attached. You can think of it as you made copies of something and so you stapled them together. So the centromere is where these two copies, sister chromatids, are attached to one another. And you have a nice image here. This is actually a microscopic image of an actual sister chromatids with the centromere. Very, very powerful microscope. And then during cell division, we'll just get started on this slide, two sister chromatids separate and move to the two new spaces that are created in the stretching of the cytoplasm of the original cell. And so they'll move to the two separate poles, and then eventually, as the story continues, into nuclei. Once the separate chromatids are in the opposite poles. They're called chromosomes. We'll pick that up again on the next one, that concept.